Hey everybody, this is Seth D. Pasquale, pharmacist and board certified sterile compounding pharmacist. I got a question the other day about a very specific thing that I had never heard anyone actually ask me. It was about hazardous spill cleanups, and the story is basically somebody was visited by the FDA and they were asked, what is your contact time for the deactivation of your drugs when you're cleaning? And of course, here in USP 800, we have, the cleaning steps that USP 800 goes through: deactivation, decontamination, disinfection, cleaning and disinfection. And there's specific agents that you're going to use for the deactivation step. And what deactivation is that it renders a compound inert or inactive. All the other things you can read on. I'm really just trying to focus on deactivation with this video. But this is all the definitions of deactivation, decontamination, cleaning, and disinfection according to USP 800. So again, here's the question. A compounder was visited by the FDA. Cleaning procedure for HDs were called into question. What is the proper contact time to deactivate your HDs? And when I got this question, I was like, I've never heard anyone ask me that question. So I, was a little perplexed. I didn't know exactly what the answer was. So I said, let me see what I can find out. And so I went on the internet and searched, and I found that there's a lot of stuff out there that, a lot of studies that basically look at particular drugs, particular agents, but they don't necessarily actually talk about contact time. I eventually got to my aha moment, but it definitely took a little while. So first, let's make a couple points. Deactivation is typically done with, and this is important, an EPA registered oxidizing agent. So a couple of oxidizing agents, for example, peroxide formulations like hydrogen peroxide, peracetic acid, sodium hypochlorite, also known as bleach. And let me just say one other thing, that there is no one proven method for deactivating all compounds. And that's right in USP Chapter 800. So there are several things that you have to kind of take into consideration when we're talking about cleaning, disinfection, deactivation, decontamination. One, the agent. Of course, you're going to select a particular agent. In this case, we're talking about deactivation, so we're going to go with our oxidizing agents. What's the concentration of those oxidizing agents? What about the pH? Does the temperature come into play? Of course, we're going to answer, hopefully by the end of this, the contact or dwell time. It also depends on the surface. And lastly, depends, of course, on the HD that you're trying to clean up. So there you go. Those are all the factors. So back to our question, what's the proper contact time to deactivate your HDs? Again, I went through a lot of studies. And most studies actually haven't looked at this particular factor. I found out of all the studies that I looked at, and I used OSHA's website. There's the link to the OSHA website that I went to. And I also went on PubMed and looked at some studies on PubMed using my own search terms, and I'll even have those here in the video too. But what I kind of settled on is just a couple different studies, and here we have one study, the pilot study evaluating the efficacy of 4 cleaning solutions and 2 types of mops in delimited areas of a floor contaminated with cyclophosphamide. So what was interesting about this study is that it actually did look at contact time, but it only looked at one drug, cyclophosphamide, but it did look at sodium hypochlorite, which is what the original compounder was actually using to clean, disinfect, decontaminate, yada yada. And then the other thing that I looked at was an SOP from the Army Public Health Center, and this is the link to each of those things. And for I referred to some other studies. Here's some of those. Pause the video if you really want to see all these other studies. They're going to go through different agents, different drugs that they tried to deactivate and what the results of those were. And these are, some more current studies. I tried to pull ones from just the last few years. So the first thing I mentioned that was really important is selecting an EPA registered agent. So let's go through just how to actually do that. And this is good knowledge to have for when selecting any kind of agent. And how the EPA divides everything up is that you can see here they have lists A through O, and it divides it up by basically what you're trying to kill. And there's the website to get to this page. Of course, you on this page, you don't really see anything for deactivation. So I went ahead and just settled on one particular agent, and that's Clorox. So then this brings you to another page that you have to go to. And with this page, you'll actually be able to get to pull up the EPA registration of whatever agent you've settled on. So again, I chose Clorox. I put it in the box here, product or alternative brand name, and I got a 34-page document, basically everything and anything you want to know about Clorox bleach. I just pulled a couple pages here for you guys to look at, but here's the directions for use, the instructions, the dilutions. Has a nice dilution table on page 32. At the bottom here is the link to this exact PDF from the EPA. But there's one other page that I think you'll find interesting. That there is one small box on one of the pages, on page, it looks like 7 out of 34. This product may be used to pre-clean or decontaminate critical or semi-critical medical devices prior to sterilization or high-level disinfection. So again, that's just kind of an interesting thing. Sort of maybe somewhat confirms that this can be used to deactivate or at the very least decontaminate surfaces. So here's what we know so far. Based on the studies that I read and looked at, deactivation requires an oxidizing agent. For simplicity purposes and to answer this compounder's particular question, I went with sodium hypochlorite. At what concentration? There is various studies that use concentrations ranging from 0.02% to 5%. And contact time was according to the studies that I saw, was 10 to 20 minutes. So again, my conclusion based off of what I could read was this. Sodium hypochlorite, concentration somewhere between 0.02 and 5%, and a 10 to 20 minute contact time. So problem solved, right? Not exactly, because there's still some factors that you have to take into consideration here. So my answer really was that you need to do your own study. And then, of course, the next question is, well, how do you do that study? So, you're going to either use sodium hypochlorite or another oxidizing agent. You're going to pick a concentration anywhere from 0.02 to 5%, and then have a 10 to 20 minute contact time. I don't know, it maybe 60 minutes. It really kind of depends on some of the things I noted here. You're going to want to take pre-samples, contaminated samples, and post-cleaning samples with varying contact times, surfaces, and varying drugs, and maybe even varying amounts of drugs as well. And I would recommend using total organic carbon analysis testing. First, to set a clean limit, that's a surface that's been clean, that's been not exposed to any kind of hazardous drug or anything. Make sure it's a clean surface. You've done a very thorough cleaning on it. Then see if your agent is able to remove the surface contaminant, and in our case, of course, is the HD, and your procedure actually works to bring the surface back to that clean limit that you've set. So some other questions. Are there labs? What labs that do TOC testing? Yes. Probably your lab that you use for your end product testing also does TOC testing. Many of them do. How do you test the surface? You're going to test the surface using sterile swabs and low TOC water. If you just watched this video that was playing here, I'll play it again. The procedure for surface sampling, this is generally what it looks like. You have a swab like that. You have low TOC water that you're going to be putting the swab into using aseptic technique, of course, not to contaminate the swab. And that's it. TOC is actually a fairly inexpensive test, so you're not going to break the bank by doing this study. And by the way, this is generally how the pharmaceutical industry is starting to do their own cleaning validations using this method of total organic carbon analysis testing. So there you have it. If you guys have any other questions, I get questions all the time from compounders, and this one was particularly interesting and I wanted to make a video about it. But if you have other questions about this or any other compounding related topic that no one else seems to have an answer for, then let me know. Hit me up either on LinkedIn, Facebook. My email's right there. I'm even willing to give out my phone number if you want to private message me and have a conversation. But yeah, that's all I got for now. I hope this was informative, and good luck with your testing, especially if the and cleaning methods, especially if the FDA comes by your place. Hopefully you'll actually have an answer. Thanks again, and as always, keep raising the bar.